Once again, I am Terry Conneran, and I am a KRAS kicker. And I am really pretty jacked up to be here today with y'all. And even though we've had a couple of little hiccups, like a hurricane, we have not really just let that go to the side, because we still have an amazing group of people that have been able to make it here from as far away as like New York and Georgia and North and South Carolina and just across the state and in Pennsylvania and what have you. So I'm happy to be here, and I'm not going to chat too much because you guys know that I do like to talk, but I want to be kicking off for our next upcoming speaker so that we can stay on target. Pardon the pun. And it is called Targeting KRAS from the Undruggable to the Druggable in Colorectal Cancer. Colorectal cancer does have a very high predominance of a KRAS mutations. And with us today, we have Dr. Daniel An, and he's a GI medical oncologist and associate professor at the Hematology Medical Oncology at Mayo Clinic. He conducts clinical and translational research focused on developing anti-cancer agents for patients with GI cancers. He is an excellent collaborator, and we are really thrilled to have him joining the KRAS conference today. And welcome to you today, Daniel. Thank you.
Great. Thank you for the kind introduction. I'm sorry I couldn't be there in person, but clinic, and I have an infant at home, so I'd rather be there. So thank you again for the opportunity to speak. So let me get my slides ready. Sure. So I actually decided to cover it a little more broadly. So actually, we're going to talk about kind of targeting KRAS across kind of GI malignancies, just to be a little bit more broader to, just because colorectal cancer is not pertinent to everybody here. So just to have a little bit more broader discussion. And so, when I think about KRAS, I kind of think about this. I remember when this article came out in 2001, so early during my med school training. And, this was the big thing that came out at that time. This Time article was talking about Gleevec with CML. And, as I continued to develop through my training and eventually through fellowship, it made me think about KRAS. And, as I quickly garnered an interest in GI malignancies, I remember reading many papers about, why isn't KRAS targetable? And, I read all these papers, and I was thinking back and saying, one day we'll eventually get there. And so I think this is just kind of a premonition of kind of where we are today, at least with KRAS. And so, in the past, we used to kind of really think about KRAS as being a prognostic biomarker, meaning we know that it's associated with bad outcomes. We know that this is the driver of many solid tumor malignancies. This is what causes cancers to be resistant to chemotherapy or treatment, and causes the cancer to spread or metastasize. And then on the left, we have predictive biomarkers, meaning are there any potential markers that are highly suggestive of response to treatment? I think with KRAS, we're kind of in the middle right now. So we're going from being a bad actor to potentially a good actor. And, as we continue to develop some of these new agents, hopefully we move more from the center to the left. And so a lot of times we hear about these terms, what we call precision medicine. So in the past, we kind of think about precision medicine as doing the testing, but what is the intent of the testing? So in every patient, we know that there are specific biomarkers, and the one of interest today is about KRAS. And are any of these potential genetic alterations of these biomarkers actionable, meaning are there any potential drugs that can potentially act on it? And when we think about actionability, I think this term below is what kind of stood out to me. So what is the appropriate therapy that leads to a disproportionate benefit? And so do certain biomarkers provide a better benefit in terms of efficacy, in terms of response for tumor shrinkage, and compared to what we have in terms of standard therapies? So really, that's kind of the crux or the goal for a lot of these targeted therapies. And so in the past, this is the way we used to think about clinical trials. I think this is a really nice depiction of kind of where we were in the past. So in the past, we used to look at drugs specifically for specific tumor types. So, they're talking about on the top, paclitaxel, which is chemotherapy for stomach cancer. We have pemetrexed in lung cancer, and we have dacarbazine in melanoma. And the way that drugs used to get approved in the past would be they would go through a phase 1 trial, so looking at the safety and tolerability. Then they would go to phase 2 to look at the potential efficacy, and then eventually they would go to phase 3 to get approved. And then as clinical trials developed, we kind of fell into this second basket where we would look at specific biomarkers within a specific tumor type. So we would look at HER2 in stomach cancer or potentially breast cancer. We'd look at ALK, which is a biomarker in lung cancer, and then we would look at BRAF and melanoma. And then they would go through the phase 1, phase 2, depending on the type of study, and potentially getting approved or conditional approval then and going to phase 3. Now here we're really at the third part. So what are these agnostic biomarkers that are not disease specific? Meaning, are there potential biomarkers like KRAS that are shared across various tumor types, where if we can show a disproportionate benefit across various solid tumors, we can potentially get an earlier approval. So meaning really in the early phase 1, where we can get a conditional or accelerated approval based off the benefit. And I think that's kind of where we are right now with KRAS, and specifically we see that with KRAS G12c. And hopefully now we have the approval in lung cancer that will eventually get it in colon cancer and other solid tumors. But I think this is really the evolution or development of where we are with clinical trials. And so I think one area of interest that I've been working a lot with is looking at what we call polo-like kinase 1 or PLK1. And so this is a target that's relevant in terms of for cell cycle signaling. So we all kind of remember from our initial days in biology, where we go through the cell cycle, where initially you have DNA synthesis, and then the cell grows, and they eventually split, and then the cell regrows again. And so PLK1, it plays a vital role in terms of the cell cycling process, and that also includes the cell cycling process in cancer cells. And we know that PLK1 targets a lot of these key points within the cell cycle, specifically between the G2 and M phase, as well as in the DNA synthesis phase. And so what they've shown in the lab is this drug onvansertib is a polo-like kinase inhibitor, and it kind of attacks KRAS from two different approaches. One, it makes the KRAS cells hypersensitive to PLK1 inhibition, and also it shows synergistic activity with chemotherapy. So 1 + 1 being 3 or 4. And so when you combine it with chemotherapy, what we've shown in the lab is that it tends to have a very synergistic effect in making chemotherapy more effective. And so this drug onvansertib is an oral inhibitor of PLK1, and it works against, again, inhibiting various parts of the cell cycle. And what we've shown in the lab is that it's shown to be synergistic with various chemotherapy agents, specifically those that are typically used in GI malignancies, so irinotecan and 5-FU, specifically in KRAS mutant cell lines, as well as KRAS mutant xenograft models. And so based off this activity, we were interested in pursuing this in the clinic. And so this was a phase 1b/2 trial that I led with Dr. Lenz at University of Southern California, where we looked specifically at combining FOLFIRI and bevacizumab, which is a typical first or second line chemotherapy combination that we would use in colon cancer with onvansertib. And so the chemotherapy was given typically how we would typically give chemotherapy in terms of the dosing and scheduling. And we would combine that with this oral agent called onvansertib, and it was specifically for patients that were found to have a KRAS mutation. The reason why we chose to combine it with this chemotherapy was based off what we saw in the lab, where we saw the synergism with chemotherapy, as well as that most patients tend to get FOLFOX in the first line, probably about 70% of patients. So we specifically looked at it in the second line setting. And the intent of our study in the initial part was to really look at the safety, but when he conducted to the phase 2 portion was to look at the response rate, meaning what is the chance that a patient will experience tumor shrinkage by our measurement criteria. And so as part of the study, in addition to looking at the potential efficacy of the drugs, we also did some correlative studies, so some studies to look at potential studies to help us assess whether or not the drugs were actually doing what we were hypothesizing they would do. So after a month, and at the time of treatment and clinical trial enrollment, at the time of cycle 1 and day 1, and then at the time of cycle 2, day 1, so a month later, we collected blood to look at circulating tumor DNA. And we kind of looked at two things. One, we wanted to look at the changes in KRAS mutant allele frequency. So essentially the changes of percentage of the KRAS mutations, as well as looking at the changes of all mutations found in the blood. And so I know that there's many of you are very familiar with what ctDNA is, but for those that are, those of you that are not, what ctDNA or circulating tumor DNA is, it's more or less what we call liquid biopsy, and it gives us a snapshot of kind of what's going on with the cancer in the whole body as a whole. And so as cells within our body grow and die, they shut off DNA, and that also includes cancer cells. And so what ctDNA looks at is the proportion of cancer DNA compared to all the DNA in our body. And so at the time, in July of this year, we enrolled a total of 50 patients at that time. And what we saw was that the response rate, meaning the percentage of patients that experienced tumor shrinkage of about 30% or more, was 35%. And so historically, when patients get second-line chemotherapy in colon cancer, the likelihood of tumor shrinkage of 30% or more, meaning a partial response or better, is about 5 to 10%. So this was about anywhere from 3 to maybe 7 times higher than what we would typically historically see. And when you look at disease control rate, meaning the percentage of patients that had disease control, where the tumors were not growing or spreading or potentially even shrinking or even disappearing, was about 94%. And then on the right here, we look at median progression-free survival. So what that means is the time that the cancer is being controlled. When we look at the tumor measurements across the clinical trial, what we saw was a median PFS of 9.2 months. And historically, we would expect to see about 4 to 5 months based off previous clinical trials. So despite this being a very small sample size, we have a very good hint of signal of the activity of what this drug can do with chemotherapy in patients with KRAS mutant colon cancer. And so as part of the correlative work, we look to see the likelihood of responses based off the KRAS mutations. And so while this is a very busy figure, what this is looking at is each individual patient that was enrolled in the study. And on the blue on the left are the patients that experienced either a complete response or partial response, meaning tumor shrinkage of 30% or more. And then on the left, on the middle, we have the yellow, which is stable disease. And on the right, we have patients who progressed. And we can see that irrespective of what type of KRAS mutation they had, whether they had G12D, which is the most predominant mutation in colon cancer, and we have G12V, 13D, et cetera, patients did experience a response of at least, did experience a clinical benefit of at least stable disease or even responses, irrespective of the KRAS mutation. And so as part of the correlative work, which we discussed earlier, is that we did collect some blood to look at the changes in the KRAS, what we call mutant allele frequency. And so through statistical considerations, we determined that the KRAS mutations needed to decrease more than 90% to be considered a meaningful change. And what we looked at was to see whether or not the changes in KRAS mutant allele frequency correlated with correlated with the radiographic response, meaning if your KRAS mutation decreases, is that associated with the likelihood that you'll see tumor shrinkage on a scan? And so what we saw was that there was a strong concordance between the two. So in patients that experienced a complete response or partial response, nearly 90% of these patients experienced a KRAS mutation decrease of at least 90%. Similarly, when you looked at the other side, we did see that if your KRAS mutation didn't decrease as much, you were unlikely to experience this shrinkage in tumors based off the scans. And so in addition to looking at the radiographic response, we are interested in also looking at the clinical response, meaning if your KRAS mutant allele frequency decreases, is that associated with you benefiting in terms of surviving the cancer, and specifically looking at preventing the cancer from growing or spreading? And so we saw that if patients did have a decrease in their KRAS mutation burden, they were likely to experience a bigger clinical benefit than those that didn't experience a decrease in their KRAS mutant allele frequency. So those two things did show a high concordance as well. And so based off these results, we are proceeding to a phase 2 study, a randomized phase 2 study, where we're looking at combining FOLFIRI bevacizumab versus FOLFIRI bevacizumab in combination with onvansertib. This is going to be a randomized phase 2 trial where I believe we're going to be enrolling close to 150 patients. And this will be open across the country, hopefully within the next several months. And so similarly, based off a lot of these activity that we saw in colon cancer, we know that KRAS mutations are very prevalent in pancreatic adenocarcinoma. So we are currently also conducting a phase 2 study in pancreas cancer, where patients that have likely received gemcitabine and nab-paclitaxel or gemcitabine, who didn't receive prior 5-FU or irinotecan or nanoliposomal irinotecan would be eligible to be enrolled in this study. So this study, again, is a second-line study for specifically in patients with KRAS mutant pancreatic ductal adenocarcinoma, where they would receive onvansertib in combination with 5-FU and Naliri. And this study is currently ongoing and enrolling across the country. And so I know that there's been a lot of interest, and I know that you've had a lot of great speakers, and we see a lot of information about KRAS G12c. And so this is a mutation that is very prevalent in GI malignancies, most predominantly in the right-sided colon cancers. Typically, we think about ambulatory cancers or right-sided colon cancers, but we can also see them in a less common incidence in pancreatic or hepatobiliary malignancies. And so this year at ESMO last month, we did get updates from both studies, from Crystal-1 as well as from CodeBreak. And so what Crystal-1 was, this was led across various institutions, including at the Mayo Clinic by my colleague, Tony Saab, who presented some of the earlier results at ASCO GI this year. They presented the updated results of patients that were enrolled in the study that received MRTX845 or adagrasib as monotherapy and in combination with cetuximab, which is an anti-EGFR monoclonal antibody. And so, as we know, KRAS G12c is prevalent in about, again, 3 to 4% of patients with colon cancer. And we know that this is associated with, again, a poor prognosis and potentially can be a biomarker suggestive of response. And so adagrasib is an oral small molecule inhibitor that inhibits KRAS G12c and has a very long half-life and suggesting potential strong efficacy. And so based off some preclinical data where we know at least preclinical and clinical data where we know that KRAS G12c inhibitor monotherapy may not be as effective due to the feedback loop mechanism. So on the right here, we can look at the MAPK pathway, looking at from KRAS, RAF, MEK, and ERK. We know that oftentimes what happens is that cancers are able to escape this inhibition or the blockage of KRAS by either activating upstream or downstream on the pathway. And so the study looked at the combination of adagrasib with cetuximab, as well as looking at adagrasib by itself. And so here's the brief schema without going into too much detail. They looked at, again, the drug as by itself, where patients got the drug twice a day, or they got it in combination with cetuximab. And so when you look at the patient baseline demographics, this is just the demographics for all the patients who were enrolled in the study. I think the main thing to really take home without going into too much detail is the prior lines of systemic therapies. So, and on average, most patients had at least 3 lines of prior therapy. So this would be their 4th line of treatment. We know typically in this scenario, at least with colon cancer, the treatment options would be kind of limited to either regorafenib or TAS-102, which provide modest responses. For some patients, they can be particularly clinically benefit, but in general, the responses are pretty modest. But some patients had up to 9 lines of prior treatment before going in the study. So some of the individuals that were enrolled in the study did receive a lot of treatment. And so here's the waterfall plot curve. So what a waterfall plot curve is to look at each individual patient that was enrolled in the study. So this is looking at patients that got adagrasib by itself, so just the KRAS G12c inhibitor. And each individual bar represents every single patient that was enrolled in the study. And in terms of the different colors, they denote what the patient experienced in terms of response, meaning did they have a partial response, meaning tumor shrinkage of 30% or more? Did they have stable disease where their cancer was controlled? Or did they unfortunately experience progressive disease where their cancer broke through grew by more than a certain criteria, in this case, 20%? So what they saw was that in all the patients enrolled in the study that received just the drug itself, they saw that the response rates were about 20%. So when you compare that to something like Stivarga or regorafenib or TAS-102, which is Lonsurf, the chances that we would exceed tumor shrinkage is about 5%, if not less. So despite patients having a lot of treatment before enrolling on this study, there was a good amount of clinical activity where nearly 20% of patients experienced tumor shrinkage of 30% or more. And so this is what we call, this is kind of like a spider plot, or tells us kind of the duration of treatment. As we can see on the bottom on the x-axis is that the time of the duration of how long patients were on treatment. We can see that several patients reached nearly that 2-year mark, and several of these are my patients in the clinic, where they did experience a significant benefit from just the drug itself, and they continue to tolerate treatment well. Similarly, here we have what we call the Kaplan-Meier curves, meaning the duration of the clinical benefit that patients received on treatment. So we have on the left, we have progression-free survival, and then on the right, we have overall survival. So the median progression-free survival was 5.6 months, and the median overall survival was 19.8 months. And so what that means into context is when patients were enrolled on the study on the right, is that on average, patients lived on average about 20 months after being enrolled in the study. And so again, this is after going through many lines of therapy. This is just really taken into consideration from the time of study enrollment. In terms of toxicities, which I think is probably the most important thing, especially when we're trying to balance quality of life as potentially the goals with treatment, we can see that the treatment was relatively well tolerated with primary gastrointestinal side effects. So diarrhea, maybe some nausea, vomiting, and some fatigue, but otherwise the treatment was well tolerated with minimal grade 3 toxicities. And so what grade 3 toxicities are those that potentially may require hospitalization, and those are based off specific criteria that look at. So in general, most of the side effects were manageable that did not require any significant intervention. So similar to the initial previous curves that we looked at with adagrasib by itself, here we have the adagrasib plus cetuximab arm, where patients receive adagrasib in combination with cetuximab. And here we can see a very impressive response rate of 46%, meaning 46% of patients experienced tumor shrinkage of 30% or more, and the disease control rate was 100%, meaning every patient that was enrolled in the study had tumor stabilization or shrinkage. And we look at any tumor shrinkage because we have specific criteria that look at before we denote it as, again, a partial response. 93% of patients did experience some tumor shrinkage of some magnitude. So again, this looks like the combination strategy may be more effective than the drug by itself. Similarly, here we look at the duration of response. So it was higher in terms of the combination rather than the monotherapy. And so here again, we look at both progression-free survival and overall survival. So without going into too much in the numbers, again, median progression-free survival was about 7 months, and median overall survival was about 13.4 months. Many of you may ask, why was the median overall survival lower? I think some of it has to do with the sample size, and the data still is relatively immature. So we'll see how this number changes over a period of time. But this is kind of what the early results that were presented in the study. So where do we go from here? Lastly here to the side effects, So in terms of the toxicities, the cetuximab can be challenging. It does add some increased side effects with cetuximab. The main side effects tend to be mucocutaneous, so skin-related toxicities, mainly with like acne, like rashes on the face, and if it's severe, it can be more than just the face, including the whole body. But otherwise, the treatment didn't, the combination didn't really add much more than just the drug itself in terms of the side effects. So the combination was in general well tolerated and looks like a very effective strategy moving forward. And so where do we go from here? So based on these early results, there is a phase 3 trial that is ongoing. So the phase 3 trial is looking at the combination of adagrasib and cetuximab in the second line setting, where patients would get randomized to receive the combination or chemotherapy. So if a patient got FOLFOX first, then they would get randomized to potentially receive FOLFIRI or vice versa. This study is a phase 3 trial that is currently ongoing across the country. And actually, it's international, so across the world. And this study is open at many sites, including the Mayo Clinic. So there are, without being biased, the reason why I'm presenting really the Mirati data is this is a study that I have a lot of experience with. There's also CodeBreak 300 as a disclaimer, which is looking at the combination of sotorasib in combination with panitumumab. And that's also a phase 3 study that's ongoing, but I haven't had any personal experience with it just because we can't open every single study, even though we want to. So that's another option as well. So what are the future directions for KRAS? And so, we've kind of briefly touched about what we call these direct inhibitors of KRAS, so G12c. There are several compounds that are being under investigation through clinical trials. We saw adagrasib, we know that we have sotorasib with Amgen, we have GDC 6036 with Genentech, as well as the Jacobi Bio compound. But shortly coming down the pipe, we will see studies involving G12D. We know that Mirati has their compound, which should be opening shortly in phase 1. And then eventually, I think things that we want to understand is how can we overcome treatment resistance? So we know eventually tumors can change and they can become resistant to these direct inhibitors of G12C or G12D potentially. And so how do we overcome these mechanisms of resistance? So not only do we have primary resistance, meaning treat patients that are not benefiting from these treatments because of either other mutations within other pathways, or what we call mutational heterogeneity, meaning that the mutation within each tumor themselves may not be expressed the same, where they may not show the same benefit, or patients may eventually develop what we call secondary resistance, meaning treatment initially works and then eventually runs its course. They've presented some of the data, and it can be related to new mutations that develop. So eventually there may be some more irreversible G12C, D, or C inhibitors that can potentially overcome these mechanisms of resistance. And then what about targeted regulation? So we know that the MAPK pathway, or in this case, well, the MAPK pathway is the pathway that KRAS sits on. We know that there are several oncogenes that are related to the activation or downstream activation of this pathway. One is through SOS, which provides the complex which KRAS G12C inhibitors work on, or SHP2 inhibitors, which leads to the downstream activation of MEK or ERK, which is downstream of RAS. So the pathway that leads to RAS activation, eventually causing tumor cells to grow. These are potential targets that are of area of interest, as well as inhibitors downstream of KRAS. So we can see that downstream of KRAS, it's kind of like going down the freeway. So you have RAS, RAF, MEK, and ERK before it goes intratumorally. We know that there are a lot of interest in looking at agents that target some of these downstream inhibitors, many of clinical trials that we've led, including MEK inhibitors or ERK inhibitors, and potential inhibitors upstream. So are there ways to block KRAS from above? So those are kind of the areas of interest that we look at when we look at direct KRAS inhibition. And then lastly, we know immunotherapy is always an area of interest, and this remains relevant also in targeting KRAS. So one potential strategy here is on so the development of cancer vaccines. So there are various different types of vaccines that are under development, peptide vaccines, messenger RNA vaccines, and then dendritic cell vaccines. And what these vaccines do is potentially promote anti-tumor activity through the stimulation of the immune system against KRAS mutant cells. Then we have B, we have what we call adoptive cell therapy. And what that is that T cells or immune cells and NK cells are removed from the body, and they're genetically engineered through viral vectors. And then they're reintroduced in the body. And these T cell receptor therapies potentially can help the body better recognize these KRAS mutant cells, to where when the cells start to grow or potentially act up, they can really inhibit their activity or potentially kill these cells. And then lastly, we have what we call TIL therapy. And so what TIL therapy is when tumors are initially removed, they isolate or remove these tumor infiltrating lymphocytes, so the immune cells that are active against the cancer cells, and they find the cells that are most active and those that are most effective, and they help generate more and more of these cells, and they reintroduce these TILs back into the patient's body. So these are potential 3 immunogenic strategies that continue to garner interest, and that are also being conducted right now through clinical trials across the country. So in summary, while KRAS remains a biomarker associated with a poor prognosis, we know that recent advances have demonstrated to be potentially a druggable mutation. So we are making progress. Future strategies look at combination strategies with chemotherapy, with immunotherapy, or other targeted therapies. And lastly, thank you for all you guys do. You guys are the reason why we continue to do all the research, and really, this is a great opportunity for me to come speak to you guys. So thank you very much.
Well, thank you so much for that. We do have a couple questions if you have a sec.
Yes, so should I just read the questions and go ahead and answer them?
I'm going to go ahead and ask you the questions and put you on the spot. So, okay, so, okay, I can't say, all of a sudden I can't spit it out. Infinetinib. Okay, let's try that.
There we go.
How come I can say it in my head and then it comes out of my mouth like just mush? Okay, can you speak to putting that with prior exposure to? Okay, can you just read the question, because I cannot seem to get these words spit out.
Not a problem. So the first question, it says, my dad has already had irinotecan. Why is this an exclusion criteria? Is there any way he can still access bevacizumab through compassionate care? So I think the question is, so firstly, the reason why this was an exclusion criteria was to look at patients that didn't have prior irinotecan, because we know that cancer cells can eventually develop secondary resistance. So what the intent of the clinical trial was to really look at patients that only had one type of chemotherapy. And the drug also showed that, at least in the lab, is that it works together with irinotecan. So specifically, they were trying to look at it in the second line setting. They are in development for looking at a first line trial, so for people who've never had treatment, but this was kind of the direction that the company wanted to go in. In terms of, they did have a compassionate use program, but this is a very small company, and so unfortunately, that program is kind of on hold because they want to make sure that there's enough drug available for patients that are currently on the study, but it could potentially reopen in the future. So I know that they know that they want to help a lot of patients, as many as possible, but for at least for now, that the compassionate or the expanded access program is kind of on hold.
Okay, that was great. I think I can spit out the next one. Okay, so as far as the MRTX 1133, is the MRTX 1133 phase 1 going to include other drugs with that, or is it just going to be a monotherapy trial?
It'll probably be both. So I know that Mirati is still under development with the drug, and I know that the plan is hopefully to open in the next several months, but when it will actually open, I couldn't tell you, but it's going to be nationwide. It'll be multi-centered. So I'm sure there will be multiple centers that should be close by to many of you patients here.
Okay, and then as far as G12S patients with colorectal cancer.
So for the onvansertib, yes. For either one of the Mirati or the Amgen studies, no, because that's specifically for G12C. But if this is in regards to the onvansertib compound, yes, they do accept all RAS or NRAS mutations.
Okay, then the last question about crossover. Oh, not the last question, but as far as crossover.
Yeah, so it said, have you seen any KRAS G12C patients go from onvansertib trial to KRAS G12C target trial? Yes. So I've had several patients that came in to get chemotherapy, specifically in the second line setting, that went on the onvansertib trial, and then once they either progressed or became intolerant for the treatment, we did enroll them to receive a G12C inhibitor, and there still looks like there's clinical benefit. So I think there are a couple things, or a couple nuances to that. One, I think, is really important about testing. So I would recommend retesting if a patient goes on to any study to make sure that they still have KRAS as the prevalent clone, just make sure that that target is still relevant. But yes, you can go to multiple studies as long as you meet the eligibility criteria. So that is not an issue.
Okay, great. And I'm just going to keep reading down G12D treatments across Europe. Any thoughts?
Yes. I don't know I don't know the answer yet about when when the Mirati compounds, MRTX 1133, when it specifically opens, but I believe it will be an international phase 1. And then eventually, as the study continues to grow, it should eventually be open in Europe as well. So that's the goal. And
actually, somebody posted the question I was thinking about, targeting EGFR and in RAS. I know it leads up down it's the top of the pathway to RAS. How does that play into treatments when it comes to colorectal cancers?
Yeah, that's a great question. So in the past, probably in the early 2000s, our thought process was that RAS in patients that had RAS activations, anti-EGFR therapies like cetuximab or panitumumab would be more effective. But as we began to understand about how these mutations work, we know that these drugs actually have a detrimental effect, or may not work or even have a detrimental effect. So if you do have a KRAS mutation, whether it's, in this case, Q61, or you have G12D or G12C, we know that these drugs aren't effective. And so we wouldn't recommend those treatments.
Okay, great. Okay, well, I appreciate your conversation with us today. And nobody gets out of here quite completely without really engaging with our audience. And how we're doing that is kind of our war cry. And, we're going to be doing a countdown to include you in it. And so if you will cooperate with us in the room, and we'll do like a countdown, like a 1, 2, 3, and all together say kick cancer's KRAS. Are you game?
Let's do it.
Okay. All right. 1, 2, 3. Kick cancer's KRAS. All right, you guys. Thank you so much for joining us today. I appreciate it. And we hope to have you back again real soon.
Anytime. Thank you so much.
Okay. Bye now.